Researchers at Karolinska Institutet have developed a new method to separate between two types of a common herpes virus HHV6 that has been linked to multiple sclerosis MS. By analyzing antibodies in the blood against the most divergent proteins of herpes virus 6A and 6B, the researchers were able to show that MS patients carry the herpes virus 6A to a greater extent than healthy individuals. The findings, published in Frontiers in Immunology, point to a role for HHV6A in the development of MS. MS is an autoimmune disease that affects the central nervous system. The cause of the disease is unclear, but one plausible explanation is a virus tricks the immune system to attack the body's own tissue. Human herpes virus 6 (HHV6) has previously been associated with MS, but in those studies, it wasn't possible to distinguish between 6A and 6B. Through virus isolation from ill individuals, researchers have been able to show that HHV6B can cause mild conditions such as roseola in children, but it has been unclear if HHV6A is the cause of any disease. According to estimates, as many as 80% of all children are infected with the HHV6 virus before 2 years of age, and many also carry protection in the form of antibodies against this particular virus for the rest of their lives. But since it hasn't been possible to tell the two variants apart post-infection, it has been difficult to say whether HHV6A or B is a risk factor for MS. In this study, however, the researchers were able to distinguish between the A and B virus by analyzing antibodies in the blood against the proteins, immediate early protein 1A and 1B, i.e. 1A and i.e. 1B, that diverge the most between the two viruses. This is a big breakthrough for both the MS and herpes virus research, says Anna Fogdell-Hahn, associate professor at the Department of Clinical Neuroscience at Karolinska Institutet and one of the study's senior authors. For one, it supports the theory that HHV6A could be a contributing factor to the development of MS. On top of that, we are now able, with this new method, to find out how common these two different types of HHV6 are, something we haven't been able to do previously. The researchers compared antibody levels in blood samples of some 8,700 megasiemens patients against more than 7,200 healthy people whose gender, date of birth, date of blood sample and other factors matched those with MS. They concluded that people with MS had a 55% higher risk of carrying antibodies against the HHV6A protein than the control group. In a subgroup of almost 500 people whose blood samples were drawn before the onset of the disease, the risk of developing MS in the future was more than doubled if they had a 6A viral infection. The younger the people were when the virus was first discovered in the blood, the higher the risk was of developing MS in the future. HHV6B, on the other hand, was not positively associated with MS. Instead, MS patients had lower levels of antibodies toward IE 1B than those without MS. Antibodies toward Epstein-Barr virus (EBV), another herpes virus that is also associated with MS, were analyzed with the same method, and the researchers were able to show that individuals affected with both viruses had an even greater risk of MS. This indicates that several virus infections could be acting jointly to increase the risk of MS. Both HHV6A and 6B can infect our brain cells, but they do it in slightly different ways. Therefore, it is now interesting to go forward and attempt to map out exactly how the viruses could affect the onset of MS, says Anna Fogdell-Hahn.